Hello. It's great to see so many people. Thanks for coming here. So it's a real pleasure to start up again with the in-person CCP4 study weekends. And we have the topic data with our subtitle from, what was it? Subtle details to big insights. So I think we've got an exciting program for you over the next couple of days. Starting with a bit of an experiment, we're going to have, after a sort of a keynote lecture, we're going to go into a panel discussion, which we've never tried before, but I think that will be very interesting. So anything to add? Other than we need to get our speaker.
Hello, my name is Maria Fando, and I'm really delighted to welcome you to our session, Integrative Structural Biology section, and I'm pleased to introduce our speaker to the session. It's Montserrat Soler-Lopez, as head of the Structural Biology Group in ECSIT. Her research is focused on the role of mitochondria in triggered Alzheimer disease, so she's gonna talk a bit more about this. And for those who online, please write to the second channel in the Slack, so we can collect your questions from there. There's yours.
Okay, can you? Yeah. So thank you very much for the introduction and for the invitation. It's an honor for me to be the first speaker of this CCP4 study weekend. So I'm going to present the work that I've been doing for the last 15 years. So yeah, stop me if you know I'm over time, because I can get carried away. So yeah, so for a number of years I've been interested in Alzheimer's disease. As you probably know, it's a fatal brain disease. It's progressively, and there's no cure for the time being. So there are two hallmarks that characterize that disease, which are there's no pointer, yeah, which are these amyloid-beta plaques, and also this is not working either, and also the tau fibrillary tangles. Those pathologies, somehow they lead to this synaptic impairment, and this leads to this neuronal loss that can reach up to 30% of the cerebral mass. So whether those amyloid beta plaques are the cause of the consequence of the disease, it has been a subject of debate for many years, because some individuals, they do have plaques, but with no cognitive impairment, whereas the opposite has also been observed. What it seems to be an evidence is that all the problems that happen in the people that suffer that terrible disease, it happens after mitochondrial dysfunction. So mitochondria, as you probably know, are the powerhouse of the cell. The cells, they are responsible for the generation of energy in form of ATP. So since neurons are highly demanded energy cells, these mitochondria are particularly important in neurons. So it has been observed that there is some decrease in the energy production in the people suffering the disease. Also, this oxidative stress associated to this decrease in energy metabolism. And also some studies, they have observed the presence of these amyloid aggregates in mitochondria. So taken together, it seems that impaired energy metabolism, it seems to be one of the first and the earliest, most consistent features in AD. So keep this in mind. So when I was in Barcelona a long time ago at the Institute for Research in Biomedicine with Patrick Alloy, we were interested in finding the connectivity among proteins under pathological conditions. So because it seems that the proteins, they tend to be more, to interact more together in complex diseases. So we tried to see if that was the case for Alzheimer's, and indeed we could see that we took just the most, the usual suspects in the disease, which are those 4 genes or proteins, and we could see that actually the connectivity degree between proteins was higher, it was 3.2 as compared to 4.7 in a random set. So meaning that indeed the proteins, they tend to be more connected in Alzheimer's disease. So this was a computational study, and then we tried to see if that could be the case doing some experiments. So what we did, this is kind of the pipeline that we applied at the IRB. So we took all the different genes that could be susceptible or affected in the disease. So from here, then we took, oh, fantastic, wonderful. Okay, thanks. So then we took all those genes and we test the interactions between the proteins by yeast to hybrid and confocal microscopy in human cells. So we found out, we identified about 250 new interactions. All of those, then we combined those new interactions with the human interactome database just to create like a more global disease-associated network. And from here, then we did some cluster analysis because this network was composed of about 6,000 nodes. Then we did the cluster analysis just to see if the proteins were grouped in particular functional modules. And indeed, that was the case. So we could identify different modules. And from here, we could elaborate some hypotheses. So going back to mitochondria, so we could see that one of the most interesting proteins that we identified in those analyses was this protein called ECSIT. So ECSIT is a chaperone that is involved in the immune response. It can also go to the nucleus to activate some transcription factors leading to apoptosis and differentiation, but also it's translocated to the mitochondria, where somehow it's involved in the production of energy. In addition to this, we found out that ECSIT is actually interacting with some of these Alzheimer's genes. So altogether, just led us to speculate that perhaps ECSIT could be the molecular link maybe in Alzheimer's pathogenesis. So since studying all together was really too ambitious for us, we decided to start from the mitochondrial function of ECSIT and see what's the role of ECSIT in the production of energy. So the production of energy is made by this extraordinary super complex of 5 different complexes, which from NADH, they manage to pump different protons until there is the complex number 5, which generates ATP. So at this point, I've moved to the ESRF to start a more molecular approach study on this. So the first complex of this respiratory change is complex 1, obviously. So this is an amazing protein complex formed by 45 different subunits. It's about 1 megadalton. So it's the entry point of the chain. So it's one of the major sources of reactive oxygen species and oxidative stress. And very interestingly, it seems that in AD, there are some problems with the assembly of these 45 proteins or subunits. So who is helping this, the assembly and the biogenesis of this complex 1? So there are at least 13 assembly factors identified so far. And it seems that those assembly factors, instead of just working independently, they assemble themselves first, just forming helper complexes that then will take care of the assembly, the different subunits. So as you can see here, there is one of these helper complex that is called the mitochondrial complex 1 assembly complex, or the MCA. And as you can see, we have our favorite protein here forming part of this complex. So at the time, that was in 2010-11, so there was very little known about ECSIT and the role in the complex 1 assembly. So there were just very few studies saying that it seems that ECSIT and NDUFAF, they were kind of co-expressed together, and the same for the other one, ACAD9. It seems that those 3 proteins somehow, they were co-expressed, and affecting one protein was affecting the expression of the other 2. And so that's why those researchers, they proposed that those 3 proteins, they would form a complex. In addition to these 3 core proteins, there would be another 2 small membrane proteins that would help to form the complete MCA complex. So then we decided just as a start to start with the core MCA complex from a molecular point of view and see how far we could get with this. So we just started to see who was interacting with whom and see if there were ECSIT was interacting with both proteins, because there were some studies speculating that maybe ECSIT could have a modular nature. So we then just tried again yeast to hybrid for this, because like this we didn't have to purify the proteins individually. And so from those experiments we could see that N-DUFAF and ECSIT, they could interact, but N-DUFAF and acanine were not interacting. And so the same for, sorry, I have to move forward. So it seems that N-DUFAF was interacting, ECSIT was interacting with N-DUFAF from more the N terminus than the C terminus. We just complemented this with some fluorescence complementary assays in human cells. And here you can see that the C terminal part of ECSIT interacts with acanine, while the N terminus doesn't. So taking all those experiments together, we kind of we could make a sketch of the interaction among the three proteins. So ECSIT indeed seems to have a modular nature. The C terminal part would interact with acanine, and then the N terminus with N-DUFAF. And very interestingly, N-DUFAF and acanine could not interact together. So meaning that ECSIT was kind of the key protein in the assembly of this complex. So then from here, then we decided to see if we could express the different modules and the different constructs. So for this, we use Darren has a platform which is called SPRI. So here you scan your protein and see if you can find different soluble domains of your protein. And as you can see, for the N terminus, we couldn't find none. Really, it was very, very insoluble. While for the C terminus, we could find different constructs that were expressing okay. Then this is just to make a long story short, we did a lot of biophysical characterization, and then we could just find, oh, sorry, yeah, so that acanine was a homodimer as previously suggested. ECSIT, we could also find kind of a dimeric form for the C terminus, and N-DUFAF was just eluting as a monomer. So from here, then we decided to study just the subcomplex made of acanine and the C terminal part of ECSIT. So we did, again, different experiments. So here you can see by gel filtration that we could really purify a nice complex. By 1D NMR, we could see that actually when we were adding acanine to ECSIT, we could see the shift of some resonant peaks, meaning that the interaction was not promiscuous, but was specific. And when we did calorimetry, we could see that actually the subcomplex was much more stable, or at least thermal stable, than acanine alone. So all this was indicating that this subcomplex was very stable in vitro, but when we did then some SANs, we did some SACs, and then also native mass spectrometry, we could see that it wasn't very clear the ratio of ECSIT and acanine. So we could see by native mass spec that actually we could have different stoichiometry. So that was very puzzling. So indicating that even if the complex seems to be stable, it had to be highly dynamic. So we weren't sure, but we decided to go ahead, and then of course it never crystallized, obviously. So then we just tried to move to Cryo-EM. And then, so before going to the Cryo-EM, I forgot to say that acanine alone is an enzyme, so it's involved in fatty acid metabolism. So it just oxidizes the fatty acid using this FAD cofactor that it's, when it's oxidized, it's yellow, as you can see here. And when after metabolizing the substrate, it becomes reduced and transparent. So the first thing that was catching our attention is that you can see the fat is yellow. The purified acanine, it's also yellow because it has the FAD bound in the oxidized state, but the subcomplex was totally transparent. So we were wondering, either the FAD is reduced or the FAD is absent from this subcomplex. So we did some UV spectroscopy just to check, because the FAD has a very typical absorption pattern. So you can see the free FAD with the two absorption peaks. It was the same for FAD bound to acanine. But as you can see for the complex, there was really no signal at all. We then check the activity by fluorescence reduction, and then we can see again ACA9 alone is active. In the presence of ECSIT, it's totally it's inactive. And then when we add external FID, it seems that once ECSIT is bound, ACA9 cannot resume its enzymatic activity. So we did again some mass spectrometry, and then you can see here that we had a nice signal for the FID bound to ACA9, the same for the free FID, but there was absolutely no signal in the subcomplex indicating that actually the FID was no longer present in ACA9 when it was bound to ECSIT. And then we did a very neat experiment. It's just, we tried to reconstitute the complex and check by gel filtration. And you can see here, so that's the peak of the complex, but when we were reconstituting, we could see at 450 nanometers, we could see that actually there was a peak coming out very late on the column. And when we did the UV spectrum of this peak, it turns to be the FID. So actually that indicated that ECSIT ejects the FID from ACA9 and also shuts down this dehydrogenase activity. So then here is, then we went to the Cryo-EM. We really had a very hard time. We had all the problems that you can imagine. It was very hard to have a good grid. We had preferential orientation, so it was really terrible. But with the great help of Irina Guce from the Institute of Structural Biology in Grenoble, we managed to have this low resolution map of the complex. So we could use the homologue of ACA9, which is called VLCAT. It had been crystallized before, so there was a predictive structure. We could really fit that ACA9 structure in our model. But then we were unable to fit ECSIT. Actually, the resolution was so low that it was very difficult to fit. So if I go back, maybe I can, yeah, so you can see, even though we had those protrusions here, that they look like maybe ECSIT was there, but the resolution was so low that we decided to just stop here and go back to the lab and try to improve the homogeneity of our complex. And for this, we decided to go back to cloning and try to find a better construct for ECSIT because it seemed to be the one causing trouble. And indeed, so this is just ACA9 alone. So we really work a lot on the purification. So in that, with these improvements, we managed to have 4 angstrom map. It's not fantastic, but at least, you know, we could have something more resolutive. We use also a single mutant that I will explain you why we use those single mutant that we were trying to impair the interaction with ECSIT. And it's in that a single mutant was gave us a kind of a higher resolution map. It was much more stable. So we managed, yeah, for the first time to have the true experimental structure of ACA9 alone. And regarding the complex, we managed then now to have a 3 angstrom resolution map. So really spending time in the lab, it's really worth when you are desperate. So as you can see here, yeah, the resolution was really high in that case. So then what we could see is that ACA9 has this kind of flexible helix at the bottom that they tend to tuck on the homodimer. So this is one protomer of ACA9, this is the other one, forming a very nice homodimer. We could really see that the FID was actually bound there. And for the subcomplex, so this is the density. We tried to fit the ACA9 model, but we could see those extra density here that could not be attributed to ACA9. And actually, we could manually build a little bit of ECSIT, indicating that, so that was actually the model of our complex. So we couldn't observe the entire C-terminal ECSIT, just this little bit that was really going, as you can see, really into the shoulder of the ACA9 homodimer. But at least we could see that there was 1 protomer of ACA9 per molecule of ECSIT. So at least the stoichiometry was 1-to-1. Voila. So then comparing ACA9 alone with ACA9 in complex with ECSIT, we could see first that ACA9, the FID was present here, but as expected, it was absent here. But then this is just the exit bit that we could observe. But if we look carefully to this part here, to this loop, I don't know if you can see, but here, this loop is closed, kind of protecting the FID to go out, while here it's completely open. So there is a kind of flip out of about 15 angstroms that just allows the FID to go away and the exit to go in. I don't know. So it seems that it's stuck. Oops. Sorry about this. Yeah. Let's see if this works now. Okay. Yeah, so as you can see, this is a close-up of the two details, so you can see the movement of this gatekeeper loop. So then AlphaFold came into our lives, and then so this is the part of exit that we can see in our Cryo-EM model, but this is actually the entire C-terminal domain of exit. So we just submitted the sequence of C-terminal exit and ACA9 into this AlphaFold complex prediction. And astonishingly, the prediction came out, it was almost exactly as the model that we had. So it predicted to this kind of binding mode of exit in that part. And when we compare the predicted part of exit with our experimental, you can see that the RMSD it's really astonishing. It's just 1 angstrom difference. The only thing that we could observe is that the AlphaFold does not predict the flipping of the gatekeeper loop. So even if AlphaFold is fantastic, it's not perfect yet. So voila. So then we went back to our old model, and this is the AlphaFold predicted complex. And as you can see, this is the part that we can see in our Cryo-EM, but this is the domain that we are unable to see in our Cryo-EM model, probably because it seems to be quite flexible here. And then that's why in our grids we couldn't trap that model here. But if you look to our old model, those protrusions that I mentioned before, it's very tempting to really put exit here. So probably this indicates that indeed that was this is the position of exit, but it can really move back and forth. So this is just to wrap up everything. So this is a movie just, so here we would have our 2 exit components. This would be ACA9. So when exit comes in, the FAD goes out, making a very compact complex. So now if we can see the closed detail, so this is what it would happen with this gatekeeper loop, that when exit approaches, the loop opens up, the FAD goes away, and exit is able to really bind to ACA9 and really embrace the ACA9 homodimer, making a very, very compact complex. So also the helices that I mentioned you before, that they tack on the ACA9 homodimer, they seem also to have a very key part in really in the formation of this complex. And all this assembly, what's the implication, the functional implication of this assembly? So well, when we have ACA9 alone, it's an enzyme that participates in fatty acid metabolism, as I said before, but it's unable to be an assembly factor of complex 1. When exit binds, the FID goes away, and then that makes ACA9 without FID, it can no longer be an enzyme for the fatty acid metabolism, but it's committed to the respiratory complex. So it becomes a complex 1 assembly factor. So exit has this very neat mechanism of just forcing ACA9 to be involved in complex 1 assembly, but just abrogating the entire enzymatic activity of ACA9, just a way to ensure that the assembly of complex 1 is properly done and there's no aberration of different fatty acid and respiratory chain, the two mitochondrial pathways that they cannot take time at the same, they cannot take time at the same time. So now if you wonder what all this has to do with Alzheimer's, so going back to the beginning, so we are also doing some cell biology. For this, we are using neuronal cells that either in wild type or overexpressing the amyloid-beta, as I mentioned before. So we could see that when we purified the mitochondria of those cells, we can see that indeed, the ones that overexpress the precursor protein, they have a high accumulation of amyloid-beta. And we could see that this has a very important effect in the activity of the complex 1. So we expected to have a shutdown activity, but it seems that it's the opposite. So in the very beginning, when those amyloids are still soluble, the effect is that they increase the complex 1 activity. And when we check the expression and the association of the assembly factors to complex 1, we can see that again is ECSIT, the one that remains associated to complex 1 when they are supposed to dissociate from complex 1 once the complex 1 is assembled. So indicating that, ECSIT remains highly associated to complex 1 even after the assembly is completed. And voila. So this is a bit the story. We have other experiments that I can't explain you today. Hopefully, maybe next time, we can see that ECSIT is really a very complicated protein. It might undergo some post-translational modifications that are also affecting the assembly with ACAT9. But I stop here. So that's the most important slide, is the people that have made this work possible. In particular, Gabriele Jacquin and Matt Jessop, they were the first doing the Cryo-EM experiments. And then was taken over by this very talented postdoc, Lindsay McGregor, that she has done all the high resolution Cryo-EM models with the invaluable help of Samira Kajwi, that is doing all the lab part. And also, my colleagues in Barcelona, all the great colleagues, in particular, Irina Guce from the IBS in Grenoble, people from EMBL that help us to do all the AlphaFold modeling, the people from the Institute of Neutrons, the ILL, that were fantastic for the SANS experiments. Also, we are collaborating with people in the Grenoble Institute of Neuroscience for all the amyloid-related experiments, INSTRUCT for the ESPRIT experiment that we did, and of course, the PSB, which is the Partnership for Structural Biology, that is the one that helped us to have a lot of platforms available for doing all the biophysical experiments. And thank you for your attention.
Thank you for amazing talk. We have one question in Slack. What the confirmation of gatekeeper loop from AlphaFold, closed or open or not?
It was closed. So the AlphaFold is not predicting this movement. So we had the same confirmation as ACAT9 alone.
Thank you. Any question from the audience here? I cannot see. Oh.